So now that we know a little bit about the neuron, what about all those supporting cells? So I'm going to mention the supporting cells of the CNS first, and then I'll mention a couple of the PNS. So supporting cells do what it says, they support. They actually make themselves into a scaffold that the neurons can attach to. So it gives the neurons a place to live, basically, a place to connect. Some of them insulate the neurons. Other types of supporting cells protect them from harmful chemicals, and in general, they promote the health of the neuron. Now, I should say that's what your book says, and that's true, but we know so much more about these supporting cells now than we ever did, way, way more. So I'm going to add a couple of things in there about them. And the first type we're going to talk about, these are in the CNS now, are the neuroglia. So neuroglia look kind of like stars, or that's what somebody thought. So they call them, that's why they call them astrocytes. Sorry, these are the first type of cells. First type of neuroglia are astrocytes because they kind of look like stars, astronomy. And they are the most abundant type of a supporting cell in the CNS. So here's a picture of it. Here's one. And up here you can see a real picture, a real photomicrograph. So they kind of do all the things I said that supporting cells do. Support and brace the neuron. So for example, this yellow thing, that's the neuron. Here's the supporting cell. And you can see it's kind of anchored to the neuron. So it gives the neuron a place to connect. It forms a barrier between the neuron and the capillaries. So here are the capillaries, and they are bringing oxygen and nutrients to the neuron, but there could be some harmful chemicals. So what the astrocyte does is puts itself between the capillaries and the neuron. See, it's between them. It's like a barrier. So it helps to screen some things out. Controls the chemical environment, absolutely. Anchors the neuron to the nutrient source. So it's bringing the capillaries close to the neuron, because remember, things have to diffuse out of one into the other. So you want them close to each other. So those are some of the things astrocytes do. Okay, so they do a lot more. So when the nervous system is developing, like in a fetus or an infant, because it's still developing, what astrocytes do is they kind of map out the pathway. The neurons don't know which way to go. Think of it that way. The astrocytes help them migrate in the right direction. That's kind of a new thing. So I would say during development, they help the neurons migrate in the right direction. They do a lot more than that. So besides helping them migrate in the right direction, they help with transmission across the synapse. Transmission. So they're helping with remember the action potential moves from the pre to the postsynaptic neuron via neurotransmitters, at least in this case. They help with that. Let's see, what else do they do? One other thing they help do is regulate the amount of neurotransmitter in the gap. So when you see here it says control the chemical environment, one of the things they control is neurotransmitters. So that's one thing they control. And there's more than that. There's like a whole list of things they do. But I just thought I'd mention that, because they're just not just supporting cells. Without them, the nervous system wouldn't develop. Okay, so those are astrocytes. Now I want to mention that neurons don't divide, and usually often cancer occurs when during division a mistake is made, and then that mistake is copied and you get excess cells. But if a cell doesn't divide or doesn't divide very much, there's often less chance of cancer forming. So cancer of neurons is kind of rare. But supporting cells, they do divide. They divide a lot. So when you hear about brain cancer, it is most often a tumor of the glial cells, the supporting cells. And so they'll call it glioma. That's just a general term, glioma, which means cancer of the support. If you wanted to be more specific, let's say it was a cancer of an astrocyte, then that would be an astrocytoma. So that's supporting cells. Another one is called a microglia, and again, this is in the CNS. So you can see a picture of it over to the right here. Spider-like. These are phagocytes. So basically they're cell eaters. They go along and they clean up dead cells or viruses and bacteria. And so they protect the nervous system. That's a big role. Protect. But we know they do more than that. So one thing we know that phagocytes do is they prune the developing nervous system. So I'm just going to write this word, prune. Developing, and NS is short for nervous system. So when the nervous system is developing, again, in a fetus or an infant or even a young child, a lot of times there are pathways, the neurons go in certain directions, but then they have too many connections. So they have too many dendrites, or they're connecting in a way they shouldn't, and the phagocytes here, the microglia, go along and snip them. Just like you would prune your garden, like you would prune a bush in your yard, they kind of do that. And that's really important. What they're finding in some children with Asperger's or autism is that the microglia didn't do a good pruning job, and they left behind too many excess pathways. So this is really important. Another type of supporting cell in the CNS is ependymal cells, and these line the cavities in the brain and the spinal cord. So we're going to go over this, but there are cavities, chambers in your brain and your spinal cord. And flowing through it is cerebrospinal fluid, or CSF. And you want to keep it moving. So if you look, you'll see these cells. And by the way, what type of tissue does this look like to you? It's on the free edge.
Epithelium?
Simple, cuboidal, right. Sometimes simple columnar, but simple cuboidal epithelium, and that's what they are. And they have these little cilia, and they're all beating in the same direction. So they create a current, and that keeps the CSF moving. Ependymal cells. Now, one other type of cell in the CNS is called an oligodendrocyte. Oligodendrocyte. And these cells produce myelin. Okay, so we're going to talk about myelin in a minute, but I'm just going to mention here that it is a fatty substance. Myelin is a fatty substance that covers the axons in the CNS. So it doesn't cover the cell body, it doesn't cover dendrites, it covers the axon. So if you look here, everything in white is the myelin. You can see, I don't know if that's the axon or the dendrites kind of poking out, and here's the cell body of the neuron. So this is kind of what it looks like. One oligodendrocyte can cover up to 60 different cells, their axons. So one oligodendrocyte can wrap around 60 different axons from different cells. And what does it do? Well, it increases the speed of the action potential down the axon. So think about it this way. Think about a lamp with a cord, right? So inside the cord is a wire, and when you plug it in, electricity goes down the wire. but you've got this plastic on the outside of the cord. The plastic is kind of like the myelin. If you took the plastic off and you stepped on the wire when the lamp was on, where would the electricity go? Into your body, right. Not into the bulb, which would be bad. So it's kind of a similar thing. You want the action potential, which remember is a movement of ions, really, to continue down that axon fast. And so if it's wrapped in something that's insulated, that's going to happen quicker. It also insulates, protects the axon, and that's why it looks white. So if it's covered in myelin, it's going to look white. Now, I should mention that in the CNS, there is unmyelinated axons. So unmyelinated doesn't mean the axons aren't covered. They absolutely are covered. It's just that they're probably covered by other supporting cells. So if it says unmyelinated, it means the axon is covered by the extensions of other supporting cells. Oh. Yes, and then it's going to appear gray, right. This is gray matter. So again, you're going to see the terms myelinated, unmyelinated in the CNS. Axons should never be bare, right? Because if that happens, you're going to get deterioration, it's going to get damaged, the nerve impulse isn't going to travel very fast. So if it is myelinated, it is the oligodendrocyte that's wrapping really thickly around it. If it's unmyelinated, it's covered by other processes, but not as well. So which one do you think the action potential would pass faster now?
Myelinated.
Right, myelinated. And that's because some axons, some parts of your nervous system, they have to communicate very quickly. So you want them to be myelinated. Some doesn't have to be that quickly. Okay, in that case, it's unmyelinated. Does that make sense to everybody? Alrighty. What about the PNS? We haven't been talking about the PNS. Well, there's supporting cells there too. They're just different. So if you look at this picture down here, here is a neuron, so the yellow cell body, and underneath the yellow processes. So you can see around the cell body here is a bunch of cells. Those are called satellite cells. And it's kind of like a football helmet around a head. That's what it reminds me of. So this protects the cell body. It protects it, regulates the environment around it. On the other hand, you have Schwann cells, and the Schwann cells cover the axon. So you'll notice the Schwann cells, each individual Schwann cell you can see is like this. And between them, there's a gap. See that? So it's not continuous on the axon. There's a little gap where the neuron is there, the axon is there. So it's not like the CNS with the oligodendrocyte where everything is wrapped. You've got these little gaps. How did that get in there? No. I don't know why that's, I didn't even notice that. No, you know what I did? I cut and pasted this, this picture, and then I cropped it, and so a little bit of the one above it. Yeah, so you should cross that out. I didn't even notice that. Fix that.
So that type of cell is in which one?
The Schwann cell?
No, the yellow one.
Oh, oligodendrocyte is part of the central nervous system. So in the brain and spinal cord, the axons of neurons are covered by that. So let me just show you real quickly. Oop. So CNS, see how they're completely covered? That's an oligodendrocyte that's doing that. But in the PNS, the axons aren't covered completely. You have a little gap there. And by that way, that little gap is called the node of Ranvier. So I'm just going to write that here. Sorry, let me write it over here because that didn't come out. Node of Ranvier. Now, I think in your book, they just call it node. But that's the eponym. Remember, an eponym is when they name it after somebody. So you'll probably see both of those. Okay, so peripheral nervous system, remember all the nerves outside the brain and spinal cord. So how do these Schwann cells wrap around the axon? How does that whole thing work? Okay, so this is just showing a little bit of how this happens. Let's see. Okay, so here's an axon. You can see in the top, the yellow thing is an axon. So this happens really during development or maybe during damage, but mostly during development. So when the nervous system is developing. So let's say that this is the axon, and I'm the Schwann cell. So the first thing the Schwann cell does is it moves itself close to the axon like this. And then it starts wrapping its body around it. See, it wraps around and around, and pretty soon that looks like a jelly roll. You guys know what a jelly roll is? Mm-hmm. Yeah. Mm-hmm. So that's what it kind of looks like. Now keep in mind that these lines here are the plasma membrane of the cell. So if you can think about it like this, let's see if I can draw this here. So in the middle here, we've got the axon. There's the axon in green. So it gets to this point where it looks like a jelly roll, and then the Schwann cell pulls the plasma membrane inward. So it's going to pull all of these in, like this. And the plasma membrane is porous. So everything that's in between here is going to go to the outside. And this is what you're going to end up with. And they show you up there too. Here's the axon. And then you get layers of plasma membrane. Okay? So these layers. So here's one plasma membrane on the outside, and here are all the others wrapped around. So when it pulled everything forward, right, all the plasma membranes came forward, and all the cytoplasm and organelles stayed to the outside. So you end up having many wrappings of plasma membrane, and this is what they call the myelin sheath. Okay, that's the myelin sheath. Just that part where you've got many plasma membranes. And they kind of show it to you here too. Now this part, from here to the outside, they call that the neurolemma. Okay, that's the neurolemma. So this picture is supposed to be this picture. So I don't know, maybe this one's better. So you can see all the wrapping around it. That's the myelin sheath. Now remember, the myelin sheath is made of many, many layers of plasma membrane. So what is there a lot of in the plasma membranes? Remember, it's a phospholipid bilayer. There's a lot of lipids. There's a lot of lipids. So when you wrap that around here, you're insulating this axon. You're insulating it because you have lipids. And it also makes it hard for ions to pass through here. They can't. They can't do that. So they call that the myelin sheath. I'm going to write this down here. It's a myelin sheath. here, remember there's a place where there's a gap between the next myelin sheath. That is called the node of Ranvier. Okay, so this is the PNS. This is how the myelin sheath forms. So let's take a look at a neuron in the PNS, which is right here. This is myelinated. So you can see the cell body up there. What are these little dots in it? Missile bodies. And what is the dot in the middle there? Right, the nucleolus. What is this blue thing? The axon, right. So here's the axon, and here's each of the the Schwann cells, each one wrapping itself around the axon. See, that's pretty good insulation, the myelin sheath. And then you've got the nodes of Ranvier. They just call it the nodes, which is a gap between the Schwann cells. Now, if you remember, the way that an action potential works, especially at this point on the axon, is you've got voltage-gated channels that open and close. And the voltage-gated channels let in sodium. And if there's enough sodium, that opens the next one and the next one. But when you've got the Schwann cell and the myelin sheath, you can't get the movement of ions across here. So the only place you find voltage-gated channels is at the node. That's the only place. And the way it kind of works is a little complicated, but I'm just going to mention that this is where you have these voltage-gated channels. So what happens is you get an action potential, right? And so you're going to get this, what happens when the action potential occurs is voltage-gated channels let in sodium. The sodium is going to flow through, along the axon, right? And that's going to open this voltage-gated channel, which lets sodium in that flows through the axon, and then it opens the next one and the next one. So it's not like you have this. Let's say this is an axon with no myelin on it, and here are the voltage-gated channels. Oops, sorry about that. So in this case, what happens is this one opens, it opens the next one, this one opens, it opens the next one, this one opens, it opens. So, see how this goes? It's like walking across the room. This is how I think of it, heel to toe. See, every time I do this, another voltage-gated channel opens, see that? So think about that. And now, look at where the myelinated one is. When it happens here, it skips from channel to channel to channel to channel. It skips. So if I skip across the room, do I get there faster than if I do the heel to toe, or slower? Faster. It's faster to skip. And so this myelin actually speeds up the action potential. They call this saltatory conduction when it skips from node to node. So the action potential skips from node to node. Okay, it's fast. Back the myelin. Okay, immune cells attack the myelin. For some reason, they think it's a bacteria or a virus. And they slowly destroy it over time. And after a while, what happens is connective tissue comes in. So if this is the myelin destroyed here, connective tissue comes in and replaces it. It's like a scar. But connective tissue does not help speed up the action potential. Actually, what happens is the axons here get destroyed as part of the process. axons get destroyed and you get this connective tissue. So what's going to happen? Let's say that this particular group of neurons, their axons, were going to the biceps brachii. Can't move your arm. Now usually there's more than one motor unit, so what happens in the beginning is there might be a little bit of weakness in that arm as motor unit after motor unit get damaged, and then pretty soon, right, can't use your arm.
Can't they get very shaky, Joe?
Yes, yes. And then it moves on to the next arm. And then the leg. And then your abdominal muscle. And then your respiratory muscle. So once that happens, you can't breathe. So they call it multiple sclerosis because this covering with connective tissue, they call that a sclerotic plaque. So they call this, sclerotic plaque. And you've got many of these plaques, so they call it multiple sclerosis. There isn't really any cure for this. There are a lot of things they can do nowadays to slow it down, depends on the person. Some people have it for years, like 50 years. Other people, boom, they get it and they don't last that long. So it just depends. But again, it kind of shows you how important this myelin sheath is.